All right, so the endoplasmic reticulum. What do we abbreviate it to? ER. The ER. Not to be confused with? Exactly. Emergency room. Oh. All right. So it's a standard abbreviation, the ER for endoplasmic reticulum. So more than 1/2 of the total membrane in many eukaryotic cells is endoplasmic reticulum. That says that whatever goes on there is very important. And why is there so much of it? Why do we have so much of it all folded? Huge surface area. That means there's something on there that needs that surface area to do whatever it does. All right. So the membrane of the ER is contiguous with the outer membrane of the nuclear envelope. And you can think about it as this system of tubules and sacs. Tubes and sacs that are kind of flattened. All right? And that forms cisternae. Now, where else do you hear the word or something related to cisternae? Cistern? Cyst? Close. Actually, so that's a kind of a different, same sound, but slightly different. The cistern on a toilet, right?
I've not seen.
You've never heard that called? Cistern on a toilet?
Never seen that.
It's the tank that holds the water.
Oh.
No?
I hate that thing.
Maybe it's not American. Maybe it's English. I don't know. I thought it was universal. All right.
It's probably called that. We just never identified it. The bowl.
The bowl. The bowl, that's what you poop and pee in. The cistern's the tank on top.
Oh, you mean the back tank?
Yes.
The flap that goes like this. Puts all the water in and out.
Yeah, we call it the tank.
The tank?
Yeah. We're American.
All right.
We make things easy.
Let's move on from that one then. Okay. So, here are the cisternae, all right? And this is the cisternal space. So these form the cisternae. Good?
Wait, so what is that?
Sorry?
What's the purpose of this?
Separation from outside, all right, from the cytoplasm. Now I'll talk about, so I did look up difference between cytosol and cytoplasm, so there is a difference. So, and I'll elaborate on that now, I guess. Think about the cytosol as the sort of jelly, watery substance inside the cell membrane that everything is suspended within, okay? The cytoplasm is everything inside the membrane, excluding the nucleus. So the cytoplasm includes all of the organelles and the other parts within the cytoplasm. If you want an analogy, imagine you've got a can of vegetable soup. Everything inside that is the soup, but the liquidy stuff that the carrots and the beef is suspended in, well, that's the liquid, the broth. That's kind of like the cytosol. Good?
Cytoplasm is everything?
Yeah, except what's in the nucleus. Okay? All right. Okay, so we've got separation then. From inside the ER, inside the cisternae, to the outside.
So it's the actual lobes of the ER, correct? It's not like some kind of surface.
The cisternae?
Yeah.
Yeah, think about it as these lobes. Okay? Those finger-like
Are
structures.
we labeling the cisternae?
No, not really, because they're not really, it's a bit like saying the table is flat. It sort of describes what their sort of shape or structure is, or the structure that's formed by the ER. Okay. Yeah? All right. So on the endoplasmic reticulum, you've got two distinct regions. All right? You've got the smooth ER, and we labeled this last time, which is the region that does not have ribosomes bound to its surface. Okay? And we call that the smooth ER. And then we've got the rough ER, and that's the part which has ribosomes bound and attached to its surface. Now, the ribosomes are able to detach and reattach. It's not fixed. In fact, most of these things inside the cell are very dynamic and can continually change and grow and move. All right. Everybody okay with the rough and smooth ER? Because they have two different functions. All right. Okay, so have a look at this diagram then. This one up here is kind of nice. It's the stylized diagram of an animal cell, and this in blue shows the ER, which again can occupy a very large volume inside the cell and can be an awful lot of the membrane inside the cell. And can you see it's like a system of tubes? There's some tubules. These are like sacs that are kind of like laying flat. Yeah. And you've got the cisternae are these kind of flaps or lobes. Does that make sense? And then you've got the lumen of the ER, or the cisternal space, yeah, which is separate to the cytoplasm or the cytosol outside.
So this is like a cut in half view, but normally
This diagram here, yeah, we've like cut right through it. And there you can see the nuclear envelope and the pores in the nuclear envelope. Okay? And this is what a transmission electron micrograph would look of, where you've sort of transitioned from rough to smooth ER. Here you can see the membrane. These are free ribosomes, and you've got all these bound ribosomes on the membrane. This is all rough ER. And then out here, the black is the membrane. No ribosomes. That's the smooth ER. Good? Okay. All right. I think that's pretty clear when you look at it,
So
the electron micrograph.
those can they change from rough to smooth or smooth to rough?
So I wouldn't say it changes from one to the other, but they can definitely increase and decrease in size. I don't know if there's a definite boundary between one and the other, but they can definitely grow and decrease in size. Ribosomes can attach and detach. Okay. All right, so let's have a look at the functions then of the smooth ER. All right, what does the smooth ER do? So one of the roles of the smooth ER is that it synthesizes lipids. It's where lipids are synthesized and made. So what is the smooth ER itself made of? Yeah, it is membrane. Yep, membrane, good. Which is mostly phospholipid, good. All right, and it's on there where there are enzymes which are able to synthesize other lipids. So what kind of lipid would this be? I know it might have been a week or 2 weeks ago, but which kind of lipid is it? Nope. Steroid, thank you. 4 fused rings. It's a steroid. The precursor of steroids are?
carbon skeleton of 4 fused rings.
So but the precursor of most steroids, what's the name of that molecule?
Oh, good question.
Cholesterol.
Cholesterol, good.
Oh,
All right.
underlined right under.
Give me an example of a steroid. Testosterone, estrogen's another one, good. All right, so testosterone, estrogen would be made on the smooth ER. So metabolizes, I'm going to go into these in a little bit more detail, by the way. It metabolizes carbohydrates. So you can imagine that there's a lot of glycogen in that area, and that's where you can metabolize carbohydrates. It's where you get drugs and other poisons that are taken into your body, either intentionally or unintentionally. It's where they get detoxified. And it's where calcium ions are stored. So that's a list of what they do, but I'm going to go over each one of these in a little bit more detail. Okay? So I know you're busily copying it down, but I'm going to flip from this slide. So synthesizes lipids. Lipids are made there. Give me 3 kinds of lipids. Fats, phospholipids, and good. Fats, phospholipids, and steroids. They're the 3 that we talked about. They're all made on the smooth ER. Okay? So, let's have a look at something like testosterone. Testosterone in males is made in the testes, and a little bit is made in the adrenal glands, which sits just on top of your kidneys. So you can imagine cells in the testes and the adrenals have a very well-developed smooth endoplasmic reticulum because they're pumping out testosterone every day. Get a little surge in the morning. Okay. What's another kind of lipid?
Wait, no. What?
Let's go with
What is that?
phospholipids. So phospholipids are produced on the smooth ER. In fact, the smooth ER essentially makes itself bigger. It grows. if it's going to make more phospholipid, itself is going to grow and extend. And so now we've got more membrane that can be used for other things in the cell. Okay? Everybody okay with that? Sure? All right. And of course, it can synthesize fats. All right? So this picture shows some adipose cell, and this big globule is mostly triglyceride molecules. All right, so much of the food you eat, when it gets digested and broken down, some of those substances can be shuttled to the smooth ER. And of course, that's going to synthesize fats for long-term fuel storage or the other things that we use fats for. So you can imagine the smooth ER in adipose cells that's going to be very active, busily synthesizing lipid, principally for energy storage. Good? So they're the one use of the smooth ER, make lipids. Okay. So another role of smooth ER is this is where you metabolize your carbohydrates and where you detoxify drugs and poisons. Ooh. So do you recognize this as being a glycogen molecule? And glycogen is a polymer of glucose. Right. So you're going to have a lot of glycogen stored in and around your smooth ER. Okay? So the minute you need glucose, that's where it's going to get broken down and glucose will get released. What are some of the main storage sites in your body for glycogen? Good. Muscle cells and liver cells. Good. That's where a lot of your glycogen is stored. So those cells are going to have extensive smooth ER where the glycogen is stored, and they can break it down quickly, release that glucose internally, which is where it might be used, especially in a contracting muscle. All right. So the smooth ER is also the site of detoxification for different drugs and poisons. So anybody know what this substance is? No? It's ethanol, alcohol. When you have a glass of wine or a beer, it's got alcohol in it. That's what it looks like, a very simple molecule. But when you consume it, your body's got to break it down. And it gets broken down, most of it, in the liver. So this is all within your blood, goes around your bloodstream. When it gets to the liver, in liver cells, they have an extensive smooth ER, and that breaks down the ethanol. Okay? All right, so what's this organ here? It's your liver. And what would that little organ there be? That's your gallbladder. It's a tiny little liver. Where did this liver come from? It was from a dog. Dog liver, okay? Not a human liver.
Have you ever had liver?
Yes. In fact, I'm going to tell you about eating liver right now.
Oh, sweet. Because all the old people don't eat liver.
So there's a nice chunk of liver sectioned through. What do you think all these are? Not disease. Blood vessels. All right? Blood vessels. That's the lumen of blood vessels through that sliced chunk of liver. It's not disease. It's a healthy looking liver. So your liver is very vascular. You have an extremely high blood flow into the liver. It's a very, very large organ. It's very high blood flow. In fact, the blood that comes from your digestive system is shunted pretty much to the liver, and the liver does an awful lot to that blood. One thing is it detoxifies the drugs and poisons. So many of the medications you take get broken down in the liver. The liver's got many, many roles, but one of them is this sort of like garbage disposal role. Breaks down the toxins before they make it to the rest of your body. Obviously, it doesn't pull them all out before they make it to the rest of your body. But so something like ethanol would be detoxified in the liver. But so let's have a look at these. So they're the blood vessels. And my mum used to make liver and bacon when we were young. She used to take liver, chop it up and fry it up with bacon. But everything tastes good with bacon, doesn't it? It does. So liver is very, very soft, except for these parts, which are very, very chewy. And I vividly remember getting the chunks and it's not going anywhere. They just pull it out. All right. But it would look very vessel-like because the vessel walls are quite strong and tough. There. But you can see how the organ is sort of divided into these little units, can't you? Yeah. And obviously these vessels will break up into smaller ones. So let's think about this. if your liver's detoxifying alcohol, right? So the smooth ER is a very dynamic part of the cell. In fact, all cells are dynamic. It can get bigger and smaller based on need. So let's just say you've never had alcohol before, and you have, I know, a glass of wine. You're probably going to feel its effects because your liver is not yet geared up to break down very much ethanol. There's an enzyme called alcohol dehydrogenase which breaks down the alcohol, and you probably don't have much of it, but it's housed on the smooth ER. So your body then says, all right, you're drinking some alcohol now, so I've got to respond by making a little bit more alcohol dehydrogenase and maybe growing my smooth endoplasmic reticulum. And that's why after maybe drinking one glass of wine for a couple of weeks, you don't feel its effects at all. Your body's responded by making more of the enzyme and increasing the size of the smooth ER in the cells that break it down. So then you go to 2 glasses of wine. Well, your body's going to make more and extend. 3 glasses of wine, make more and extend, but there's a limit to what your body can do. And then, of course, you start to damage the tissues if you drink too much. Yeah.
So when, say, like you're an alcoholic, does that smooth ER start getting really damaged where it can't start producing that?
So it will get big and it will try and deal with it, but then, of course, you're going to suffer the toxic effects of the ethanol because there's only, your body can only do so much. Yeah. It's just like an overdose of any drug or any poison. Yeah. Your body can break down some, but there's a limit to what it can do, and then you start to damage, and I guess that must impair the ability of the liver to break down ethanol.
So say someone's trying a pain medication for the first time and it affects the liver right away. What is that causing? Like, how does that work?
So I don't know.
It's the first time taking the pain medication.
Yeah, so, I mean, this is your garbage disposal organ, right? And it's the biggest organ that receives the blood from your digestive system. So anything you take that's absorbed by your digestive system is going to go into the liver, and the liver's going to try and break these foreign chemicals down. So if some medications, the liver's going to try and break it down and maybe not know what to do, not know how to break it down, or it might be toxic to the liver. So many blood tests you do, they're looking for the presence of, say, liver enzymes, because the liver's become damaged, the cells have broken open, and those enzymes have been released into the blood, and there's unnaturally high levels of them. Right? But I would imagine that many medications that you're going to develop a tolerance for them over time in the same way you do for ethanol. Yeah.
So if you don't drink a lot, like say you drink like 1 glass of wine every like 2 weeks, like
Yeah.
would it get bigger and then get smaller? Or would
So
it,
I don't know.
It gets bigger,
That's a good question. I don't know. I don't know how quickly it will respond. I don't know the answer to that. I don't. It's a good experiment to do, especially if it involves wine, right? Yeah.
So like all the toxins or anything that it kind of filters out, where does that stuff go?
All right. So the breakdown products of it can go in a lot of different places.
They don't
All right.
stay in the liver though?
Sorry.
Do they stay in the liver?
They
at all?
shouldn't. I mean,
Okay,
some of them get stored in your body, and some of them don't leave. All right. There's some that your body's unable to get rid of, or at least they get rid of them very, very slowly. All right. So there's different places that they can be disposed of, but your urine is a big one.
Okay.
All right. Some of them in the sweat and so on. Okay. All right. Some things that the liver metabolizes, it metabolizes into substances a lot more toxic than the original substance. Right. So methanol, wood alcohol, which is similar to ethanol, is metabolized into formaldehyde. That's extremely toxic, and that has a huge damaging effect on your liver. All right. All right. Good. Whole new appreciation for the smooth endoplasmic reticulum now, right? Yep. And your smooth endoplasmic reticulum also stores calcium, which is extremely important in muscle contraction. So there's loads of calcium stored. When your muscle cells need to contract, calcium leaves the smooth ER, and then it rushes back in. So there's a constant flux of calcium in and out, and it's related to muscle contraction. So you can imagine muscle cells have a very extensive smooth ER to store the calcium required for muscle contraction. Good?
So calcium is on the smooth ER?
Stored within it.
Oh.
Okay, so that's a variety of roles and functions of the smooth ER. A lot goes on, and there's a lot of enzymes in that region and embedded within the membrane. Okay. So now let's look at the rough ER then. What does the rough ER do? Rough endoplasmic reticulum. So it's got ribosomes bound to it. And this is where proteins are made that are destined to be secreted from the cell. So any proteins that need to be secreted from the cell, that's where they're made. So think about this. Many tissues and organs in your body produce proteins that are released and have an effect on another part of your body. So a good example of that would be your pancreas, right? You've got cells in your pancreas that produce insulin, and they're produced on ribosomes on the rough ER. They're then packaged into little transport vesicles. I'll elaborate on this. And then they're transported, released from the pancreatic cells into your bloodstream, and they have an impact on other cells in your body. Okay? So, proteins that are destined to be secreted from the cell are made on the rough ER. Now that's in contrast to proteins that are made in the cell but used in the cell and not secreted. Those proteins are made on the unbound ribosomes. All right? All right. So, the polypeptides are made on ribosomes that are embedded in the ER membrane, and they're kind of threaded through pores into the lumen of the ER. So they're threaded through pores, and the proteins find themselves in the lumen of the ER, in there. I'll show you a diagram that will help sort of visualize what's going on really close up. But it's in the cytosol, yeah, where all the amino acids are. And so you can imagine, if I'll draw a slightly bigger diagram of that. So here's the lumen of the ER, here's the cytosol. Okay? You've got the ribosomes that are embedded, small unit, large subunit. Yep. Amino acids are stitched together. Imagine if all of these are amino acids. This is where they're joined into a polypeptide. Then the polypeptide makes its way through a pore into the lumen of the ER, and then that polypeptide will fold into its three-dimensional shape. Okay? So those proteins are accumulating in the lumen of the ER. Okay?
And what do they go through, that thing that's like this?
Well, that's a ribosome. They go through a little pore. I'll show you a better diagram of it.
I think you mentioned it before, so now it's making more sense.
Okay. So once the proteins are inside the lumen, carbohydrates molecules are often attached to the proteins. And when a carbohydrate and a protein molecule are bound together, we call those glycoproteins. Glyco for the carbohydrate part and protein for the protein part. So very often carbohydrates are bonded to the proteins, covalently bonded, and we call those glycoproteins. Now, proteins that are destined to be secreted, they then get packaged into transport vesicles. And it kind of works like this. Imagine here's a bunch of protein molecules. I've exaggerated their size maybe a little bit. So there's our protein molecules. Now I'm going to add my little carbohydrates to them, just maybe a little bit. Okay? Now what happens is the ER kind of does this. And all those proteins get moved in there. Okay? And then this can pinch off. I'll draw sort of a more advanced version of it. And eventually it will form its own little bubble, a transport vesicle. Yep.
Do those turn into like the motor proteins that like travel along that are being pulled?
So these don't turn into motor proteins, but these are what the motor proteins are dragging along to move them. And I know it looked like they were moving slowly, but man, they can truck around pretty fast.
And that's done through ATP and ADP, correct?
It is. That supplies the energy to make the motor protein change its shape. Yep.
So are those vesicles, whatever?
Vesicles.
Are they like unattached from it and then come and then like merge merge with it, or do they like kind of?
Well, this would be where we're producing one,
So
because
it's produced there. It's not like something that just comes and is like, hey, you got something for me to pick up?
exactly. So you can think about
It's like a cab or something.
it's the membrane that's already there, and it will form that little pocket.
Okay, so that makes
Yep. It's vesicles can fuse with it,
sense. Okay.
but
Some
in
are.
if it's the traffic is going to be mostly one way away,
Okay.
all right? Because these proteins are produced, then they need to be packaged and shipped somewhere else. Okay. All right. And the rough ER is also this membrane factory for the cell. So if you can imagine, if we've got these vesicles budding off all the time, the rough ER would just get smaller and smaller and smaller, wouldn't it? But it has to keep expanding itself, making more membrane to compensate for the vesicles that are lost. And then the vesicles join to other parts of the cell. And so they can then expand other membranous parts of the cell. Okay? All right, so this diagram hopefully will sort of help make sense what we just looked at. All right, so imagine this here is the ER membrane. Okay? There's a little pore. See, there's a little pore in it. A ribosome comes along, attaches directly over the pore. There's all amino acids up here in the cytosol. They get joined together, and each one of these little beads is an amino acid. This is a growing polypeptide chain. So the polypeptide chain is thread through that pore into the lumen of the ER. It will then fold into its correct shape. Maybe a carbohydrate will be attached to it to form a glycoprotein, and then these will all get packaged into a vesicle
It's like a treble clef.
in this way that I showed you. It does a little bit. All right. In contrast, a free ribosome here is going to produce its polypeptides, and this protein might have a role inside the cell. So it may not need to be packaged in a vesicle to be transported. Everybody okay with that? Good? All right, so we're going to leave the ER now, go on to our next organelle, the Golgi apparatus. So I'm going to draw the Golgi. All right, so a bunch more to label then. We've got the Golgi apparatus. We've got these transport vesicles. And I'll label that in a sec. We'll talk about the Golgi. So, everybody okay with the Golgi apparatus? Added it, labeled it, and transport vesicles. So it bears a resemblance to the endoplasmic reticulum. It's these sort of flattened, stacked sacs that also form cisternae. Okay. Transport vesicles from the ER move from the ER to the Golgi. So substances that are made on the ER are transported to the Golgi apparatus. And there's directionality. in the movement of substances through the Golgi apparatus. So there's a side which receives the transport vesicles, all right, a receiving side, and a side where they usually move away from the Golgi to somewhere else, for example, to the cell membrane to be released or secreted. All right. So the Golgi apparatus has a cis and a trans face. The cis is the receiving face, and the trans is the away from face. And substances kind of make their way through the Golgi in that sort of direction. And sometimes they can be sort of budded off on the edge. So when the glycoproteins produced on the rough ER arrive at the Golgi apparatus, here's what the Golgi apparatus does with them. So you've got all of your proteins inside these vesicles. And the vesicles, the membranes fuse. And so the vesicular content will be released to the inside of the Golgi. And the Golgi does a lot. One thing it does is it modifies those proteins or glycoproteins. And it modifies them in a whole variety of ways, but they can be chemically modified. Okay? Some molecules are manufactured and produced on the Golgi apparatus. That's where they're made. And sometimes things can be stored there. Sometimes molecules can be sorted. And then again, packaged into transport vesicles to be shipped away to other places.
Is the Golgi made up of the same thing as the smoothie R? Is that why it's able to kind of like...
It's also made of membrane, yep. If I've drawn it in blue, it's plasma membrane, yep. All right, so here's a better diagram than what I can draw to give you an idea of what it looks like. And you can see all the vesicles sort of coming in, fusing, dropping off their contents, maybe moving through, new vesicles being produced and then being released from the shipping side. Yep. So obviously you've got the diagram just there, and here's what it looks like when you look at a TEM of the Golgi apparatus. Vesicles arriving, releasing their contents, modification, storing, sorting, shipping, something's being made, moving through the Golgi, then being packaged into vesicles and being shipped out away from the Golgi.
So what kind of modifications is it?
So, a whole variety, like an enormous array. Like think about up here when the protein is made, then carbohydrates are attached to them. So maybe the carbohydrates are modified, maybe more carbohydrates are attached. But the modifications are huge to the different proteins. Okay? Anytime a protein has something else other than amino acids on, it could be added in the Golgi. Okay? Sometimes more than two polypeptide chains are joined together to form a functional protein in the Golgi, for example. All right, so here's kind of a pretty cool computer-generated sort of three-dimensional rendition of what the Golgi apparatus looks like. And that's all plasma membrane. Get a picture of what it looks like, yeah? All right, so this next one, I do not know for the life of me how they take some of these videos because they're on such a phenomenally small scale. All right, so this is going to show you transport to and from, come back. Transport to and from the Golgi. See things coming and going. To be able to get it that small and to be able to isolate it and separate it from the other components in the cell, I don't know how they do it. All right, so now we're going to look at lysosomes. So I'm going to label some of these transport vesicles. In fact, what I'm going to do is I'm going to fill them with little black dots to distinguish them from the other vesicles. And I'm going to label them lysosomes. So lysosomes. I want you to think about lysosomes as just being a very specialized type of vesicle. Right, they're a little vesicle. What makes them lysosomes is what they contain. So there's this little membranous sac of hydrolytic enzymes. That's enzymes that are able to undergo hydrolysis reactions. What do hydrolysis reactions do? Good. Breaks down a polymer into monomers. By doing so, it needs to split a water molecule so that one monomer receives the hydroxyl and the other one the hydrogen. All right, so you can imagine these lysosomes as being full of hydrolytic enzymes that can break down all the macromolecules in the cell. What do you think about that? These contain hydrolytic enzymes, enzymes capable of breaking down every macromolecule in the cell. Yeah. Doesn't that sound kind of dangerous?
Yeah.
Yeah? All right.
So could they shut down your whole body then if they can break down your cells?
So I guess they've got that potential, but thank goodness they're locked up inside the membrane sac. Now, why don't they digest the sac itself, since it's made of membrane, which is plasma membrane, and there are enzymes in there capable of breaking down plasma membrane? They're actually kind of not quite coated, but the interior of that plasma membrane has substances, and I'm not sure what they are, but they sort of are immune to the lysosomal enzymes. They bud off from the Golgi, all right? So the proteins are going to be produced on ribosomes on the rough ER, okay? And then those proteins are going to be put into a vesicle. They're going to be shipped to the Golgi. They're going to be modified into functional lysosomes, and then they'll bud off from the Golgi, okay? All right, so inside a lysosome, the pH is about pH 5. And that's an optimal pH for those hydrolytic enzymes. They work best at pH 5. Okay, so if they are released into the cell, the cytosol and cytoplasm inside the cell is about pH 7, so they don't work quite as well. All right, so optimum is about pH 5. All right, good. So the enzymes themselves are made on the rough ER, packaged into vesicles, transported to the Golgi. Golgi is going to do all sorts of modification, presumably make sure the pH is at pH 5, package those hydrolytic enzymes into vesicles again, and they are then lysosomes because of what they contain.
And then what's the purpose of them?
We'll get to that in a sec. It's a good question. They've got a whole variety of different roles. They've got one role really, but they can accomplish a lot of different things with that one role. They contain enzymes that break down macromolecules. That's what they do, break down macromolecules, but we're going to look at how they're used. Okay. All right, so some cells can engulf another cell by something called phagocytosis. All right, and I'll draw a quick picture of kind of how that works. So think about an organism like an amoeba. It's sort of the classic example. You saw amoeba in one of the early labs in the semester. Yep. So an amoeba's kind of doesn't really have a definite shape, does it? There's the nucleus. But amoeba eat other organisms by engulfing them. So the amoeba kind of does this. If that's the cell that they're going to feed upon, yeah, they extend their arm. It's called a pseudopodium, and by something called phagocytosis, they'll engulf that. So the next stage would sort of look like this. There's the cell they just engulfed. Yeah, and the next stage would kind of look like this. Good? So that would be a little food vacuole. There's a cell that it just engulfed. We would call that a little food vacuole. We'll talk about vacuoles in a moment. But what does the amoeba do with it now? How does it break it down? It floods it with lysosomes. All right? So little lysosomes then fuse with that vacuole, release their highly destructive enzymes, and it breaks down whatever food is in that little vacuole.
It's kind of like the Lysol of the cell. Okay.
It's well, think about it as like you release all your digestive enzymes into your digestive tract to break down the macromolecules. What this is doing, it's releasing those hydrolytic enzymes from lysosomes into that little food vacuole. To break this down, break whatever organism it just ate, yeah, break it down into monomers, and it absorbs the monomers and it builds them up to whatever polymers it needs. Does that make sense? Yeah? So one role of these lysosomes then is to digest food that other organisms can engulf. Okay?
Does the lysosome, does it have a definite shape?
Yeah, they're just little sacs. So I've drawn them as round. I mean, they kind of like these, you can almost think about it as like a bubble, not completely spherical, but largely. They're little spheres. All right, so some cells can engulf other cells, yeah, by a process called phagocytosis, which I'll talk about a bit later. That forms a food vacuole. So lysosomes then fuse with that food vacuole, and those hydrolytic enzymes can digest whatever's in the food vacuole. Now, it doesn't have to be the cell, another organism. It can be a small food particle or something like that. It doesn't have to be a whole cell. So this is a really good graphic. I think it helps explain it. So here's some food. I don't know what it could be. All right, any sort of food. So that food might be brought into the cell, all right, by phagocytosis. So now we've got a little food vacuole, right? This is membrane. There's food in there. Well, the organism's got to digest it. It's got to break down the macromolecules in the food. So there's a lysosome with those digestive enzymes inside. If they fuse together, the digestive enzyme is going to break down that food, and then the monomers can be released into the rest of the cell and built up to whatever polymers the cell needs. Yep.
Sorry, I have another question.
Yeah.
So if the lysosome attaches to the food or the other...
They just kind of fuse together.
Okay.
Have you ever seen like two bubbles, when you've blown bubbles, and two bubbles come together and they fuse into one big one?
Yes.
That's kind of what happens when these lysosomes and the food vacuoles fuse.
How does the lysosome not eat through the other cell?
So,
It's no longer contained.
so why doesn't it eat through this vacuole here?
Yes.
So there's got to be some of that digestion going on. There will be some. So remember, the pH is an optimal pH of about 5, right? So as soon as they fuse, that pH presumably is still at 5, and they're going to be breaking them down. But there's got to be processes that are going to inactivate or deactivate the lysosomal enzymes. But they're going to do some destruction to these food vacuoles unless they're coated with the same, or unless they have the same composition as their own vacuole. Okay? And there are other mechanisms the cell's got to kind of regulate enzyme action. Do you remember in lab I talked about enzyme helper molecules? So you can release those and the enzymes work well, and then you can take them away and the enzymes kind of stop working. So I don't know if that's at play with the lysosomal enzymes. I would imagine it would be. We're going to look at lab later in the semester where magnesium ions are really important to the functioning of an enzyme. If you take them away, it stops working. If you give them, they start to work. So pH is one way that you can sort of regulate the lysosomal enzymes, but there might be some coenzymes or cofactors. It's a good question. All right, and now have a look up here. So the lysosomes tend to stain very darkly on transmission electron micrographs. All right. All right, can anybody guess what on earth might be going on there? It's a scanning electron micrograph that's been coloured. So there is some phagocytosis. This is actually a bacterial cell. All right, I think it's a Staphylococcus. And it's being engulfed by a white blood cell. All right, white blood cells engulfing it. And your immune system is amazing. It's very complicated. But at one level, here's what's going on. That white blood cell is engulfing the bacterial cell. So now we've got a situation similar to this, haven't we? A cell that's just engulfed another cell. Your white blood cells are going to flood that vacuole with the bacteria in with lysosomes. And the lysosomal enzymes are going to presumably break down and destroy the bacterial cell. So lysosomes are really important in white blood cells in your immune response. So that's one role of lysosomes. Break down food particles, kill bacteria in white blood cells. Same sort of concept. Here's another role. Lysosomes are able to recycle many of the macromolecules in your cell. So you have a lot of macromolecules in your cell. Some of them are needed right now, and then sometimes they're not needed. They're needed maybe in one instance and not another. Sometimes the organelles in your cell are needed, and sometimes they're not needed. All right? So you can recycle the chemicals that the organelles are made of, and you can recycle the macromolecules, so you can use the monomers that they're made of. Now that process is called autophagy. Auto means self, phagy means eating, self-eating. So you can use the lysosomes then to recycle things in the cell. Half of your liver cells' macromolecules are recycled each week, broken down into their monomers and build up into something different. Okay? So what kind of macromolecules are going to be recycled? Carbohydrates. What else? Proteins, lipids, and nucleic acids. All right. So they all get recycled within the cell, and lysosomes have a very important role in that.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the RUB-ER and moved through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Yep. Can you see the flow of things then? Yep. Now that's a lysosome, and it's ready to do whatever it needs to do. All right. So this little graphic then, this is a really nice electron micrograph. Here we've got a mitochondrial fragment. All right, so you've got mitochondria in your cells, and sometimes they get damaged and they don't function anymore. So they need to be recycled. This is a peroxisome fragment. A peroxisome is another organelle which is involved in breaking down substances, accepting their hydrogen ions, making hydrogen peroxide, which then very rapidly gets broken down because it's quite toxic. But sometimes organelles don't function anymore and they need to be recycled. So they can be fused with a lysosome. The lysosome will break them down, and all those macromolecule monomers can then be reused for something else. So here's the electron micrograph, and here's kind of the diagram of what's going on. Here we've got a vesicle containing mitochondria and peroxisome. Fuse it with a lysosome, the enzymes break it down, and those can be used for something different. What do you do when something gets damaged? Most of the time it doesn't get fixed, let me tell you.
You mean like in real life or in our bodies?
In real life.
Oh, okay.
Stuff at home, your refrigerator breaks, what do? Your watch breaks, you toss it away, right? Your cell is so good at recycling and reusing things. It's an ethic that we need to adopt, really, right? All right, so here's another example then of the role of lysosomes. Anybody know what this is? It's a very young embryo, the hand of a very young embryo, right? It's an ultrasound, colorized ultrasound. Can't you see the little digits there yet? Now, here we've got some developing digits there, and there you've got skin between them. Well, that skin's got to go away. Those cells have got to go away, so does that tissue. So cells in that little webbing there, early in embryonic development, they get broken down by lysosomes, and those molecules are reabsorbed into the developing baby and used to make other structures within the baby. So these cells literally auto-destruct, go through programmed auto-destruction. Yep. Here's another good example. There's a tadpole. What happens to the tail on a tadpole when it turns into a frog? It gets reabsorbed, right? Well, it can't just like reabsorb a tail, but the tail cells undergo self-destruction, and it's the molecules that then get reabsorbed, reassembled to make legs, for example, or lungs. Okay? And lysosomes have an important role in all of that. Good. Have you got a good idea what lysosomes do then? Yeah. Good. All right. So now we're going to move on to vacuoles. So let's add another label to the diagram. I'm going to draw a vacuole like this, and maybe another one over there. So vacuoles can be fairly big. So you can almost think about these as being like very large vesicles. They're very large bags or sacs that are bounded by membrane. All right? But the key thing is that they're very large. All right, large vesicles. And they're derived from the ER and the Golgi apparatus. So the membrane that encloses them comes from the ER and the Golgi. And they have a wide array of functions depending on the cell that they're in. I know we're focusing on animal cells, but plants have all of the organelles we've talked about so far. And they also have vacuoles. So, a vacuole can be a food vacuole formed by the process of phagocytosis that we just talked about. So I could have a food particle in there. Or if I'm an amoeba, I could have a whole other protist in there. Or maybe a bacterial cell, if that's what I'm feeding on. All right, so it could be a food vacuole containing food. And if it's a food vacuole, what is the organism going to flood that vacuole with? Yep, lysosomal enzymes, good. There's something called a contractile vacuole, which some organisms have. A lot of protists have something called a contractile vacuole. Now I'll show you a video of how these work in a moment, but a contractile vacuole essentially does this. It starts off being big and it's filled with water. And then it contracts and it squeezes all of the water outside the cell. Now, do you remember the lab we did where we looked at the movement of water in and out of cells? So if I've got a protist like this amoeba or a paramecium, for example, and this is in fresh water, where is the ion concentration going to be the greatest? Inside the cell or outside? Inside. So what direction is water going to move? Water's going to move inside by osmosis over the entire area. Water's going to be flooding in. Now, if it did that continually, it would just get bigger and burst, right? So what the amoeba is able to do, it's able to move all of that water to a vacuole. So there's the vacuole, tiny, well, it's filling up with water. Okay, so the water moves into a vacuole. Starts off small, and then as more water's pumped in, it ends up very big, right? And then that vacuole squeezes all the water to the outside. It's literally like a pump. Good? Contractile vacuole.
So is it connected to the cell membrane?
Yes. And then you've got something called a central vacuole. And lots of mature plant cells have central vacuoles. And they can be very, very large, occupy a very large volume of the plant cell. And they're mainly designed as little storage vacuoles. They hold a variety of organic compounds and water, for example. All right. So vacuoles then have a variety of different roles. They're just like big holding tanks for something. All right, so there's an amoeba, it's a protist, and there you've got its contractile vacuole, and there you've got a food vacuole. Good, food vacuole contains food, obviously, and this contractile vacuole contains water. Yeah. So here's a plant cell, diagram of a mature plant cell. Look how big the central vacuole can be. Yeah, it can be huge. There's a transmission electron micrograph one, and again, it can be used maybe to store water or other organic molecules. All right, so it's hard to see on this little video clip, but here's a paramecium. I think you saw these in the lab. See these little hair-like structures on the outside that cover its body? They're cilia, yeah? All right, so in this region here, you're going to see something get big and small and big and small. It happens quite quickly, but just look. Water's big, see that? Water gets shunted in, then it gets emptied. It's filling up with water. Fill, 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 pump that water in, and then it empties. Good. You can see that when we have big paramecium in the protist lab. You can see that happen. It's kind of cool. But you've got to slow them down. They don't stop swimming. It's tough. All right. Good still? Okay. So now I just want to introduce you to the endomembrane system. All right. Endo means inside, membrane, you know what membranes are, the endomembrane system. So the endomembrane system consists of these cell parts that we've already talked about. So the endomembrane system consists of the nuclear envelope. Well, there's our nuclear envelope. It consists of the endoplasmic reticulum, both the rough and the smooth. The Golgi apparatus. Lysosomes and vacuoles and the plasma membrane. Oh, sorry. Okay. All of those form the endomembrane system of the cell. Now, some of those parts of the endomembrane system are directly connected, and some of them are connected via transport vesicles. So I want you to try and figure out or look at your diagram and which are physically connected, which parts of the endomembrane system are physically connected with each other and which are connected via transport vesicles? So what's physically connected to what?
Nuclear envelope to the ER.
Yep, they're physically connected. All right. What about Golgi? Is it physically connected to any of the other parts?
No.
No. How is it connected?
Transport.
By transport vesicles. All right, good. Good. Nuclear envelope, ER, transport vesicles, Golgi, plasma membrane, and vacuoles. All part of the endomembrane system. Now, it's constantly changing in size and composition and thickness. It's under constant flux. The ER gets bigger and it might get smaller. The Golgi might get bigger and it might get smaller. You might have more vesicles at one point and less than another time. And the composition of them change. Which enzymes are present, which proteins are present, what's being made, what's being disposed of. It's in constant flux. Okay? What kind of things do you think it responds to? Well, in you, maybe a little bit. In some organisms, probably. I'm just thinking about you and your lifestyle. What's going to quite radically change this in some cases? Your diet, the food you eat. Absolutely, the food you eat. Okay? If you live on Pepsi and pizza, it's going to look very different to someone who eats a lot of lettuce. All right? Okay, exercise, that's another one that's going to change it. All right? If you're contracting a lot of muscles, you're probably going to have quite big, smooth ER to let you do that. Okay? Age is another thing. Okay? Medications that you're on. So a lot of things affect this, and it's in constant flux, responding and trying to meet your needs. All right, so there's some of the reasons why. So here's a diagram, shows you all parts of the endomembrane system, nuclear envelope, the ER, the vesicles, the Golgi, plasma membrane, and lysosomes. Good? And you can see flow is mostly in one direction, isn't it? The way things flow. All right, I think this diagram shows it a little better, although this diagram is not in your book. Okay. All right. So, hang on, we're not done. You thought we were done. Not done yet. We'll start on the mitochondria. All right, so I want to add one more organelle to your diagram. I want to add the mitochondria, okay? Again, I'm going to draw it in blue because it's membrane. I'm going to put one up here. And I'll put another one down here. Okay, so there's two mitochondria. Singular is mitochondrion. Okay. And we'll talk about them next time. I just wanted to get them labelled on your diagram. But these have a role in chemical energy conversion. They simply shuttle chemical energy from one molecule to another. All right. Good.